Dear friend, most welcome in my channel, and we are again coming some new things, new topics. We are going to differentiate bacteria on the basis of their staining method and cell wall structures. We can see this Facebook page where you can find more detail about bacterial informations, all the information about related to microbes. This is my channel. So my friends, we can go in this bacterial staining process and which type of bacteria, gram positive, gram negative, we are see in the later sections. First things, who discovered this staining process, how the bacterial cell wall is differ from one bacteria to another bacteria. This is the main things of this staining process method, because most of the bacteria are not harmed or there are harms. Why? The cell wall structure, gram stainings, and the scientist Hans Christian Johnson Grams in 1884 space. This is the Denmark scientist. They can discover this staining process. This is a gram staining process on the basis of cell wall morphology. It is some cell wall differentiation. It's a negative and positive bacteria. So we will later section we will describe all the details about this. So some bacteria using by staining, colorizing agent, decolorizing agent, contrast stain, so we can find out color of the bacteria. This is the cell wall of bacteria, positive and negative bacteria. You can see there is a lot of difference in their structures, morphology. So now we will start our things. So my friends, we are coming to staining process, how to stain these bacteria, how can differentiate. You will find out this information in details in this Facebook channel, Facebook page, where you will go and search Journal of Research in Microbes. So you will see a lot of other things also in these pages. So first of things, which type of stain we are going to use during the differentiation of bacteria. This is the primary stain, and this is called as the crystal violet stains. How to make this crystal violet stains, you can just read here. Crystal violet 2 grams ethanols 20 ml. You can mix them, and this is the one solution in one bottles, and second solution, second bottle, B solution, you just mark in your bottles A and B. Other things you just like ammonium oxalate 0.8 grams and distilled water 80 ml. Properly you mix them, and this solution will filter and store in the colored bottles. I want to ask you why we need to go for the colored bottle for most of the solution in storage. You just comment on the section in below. Second solutions we need to be make the ready before we go. Staining process is the gram iodine. This is called moderants. What the role of this iodine, I will tell you later the details about these chemicals, different roles during this staining process. So before we are telling details, we need to be prepare all these informations with these pages. Like this pages, you will go for the reading iodine 1 grams and potassium iodide 2 grams, distilled waters up to 100 ml that we can mix to, and also colored bottles we can store in the fridge. Decolorization solution, just like which type of decolorization, how can decolorize anything. You know that these are the alcoholic compound, because they are dehydrating agent. They can dehydrate the solution and they can make the decolorization of anything. So they can be remove the water from the any shells, any living things. So how can you make this decolorizer? You can add 95% of ethanols 50 ml and acetone 50-50, 1% acetone, like you can mix and you can store in the bottles. Counter staining, why we need to counter stain, I will tell you later. This is also making by using saffron O 0.34 grams and absolute alcohol 10 ml and remaining 90% ml of the distilled water and also colored bottles. Now the roles of the crystal violet, how can they affect the membranes of the bacteria, the anything just like that. The crystal violet is chemicals that can be crystal violet and chloride solution. When they can be in solutions, they will separate in these type of ions, and these ions penetrate the cell walls of the bacteria, and the cell walls of bacteria is the main thing to differentiate these bacteria, either they are positive or negative gram. So these gram positive, gram negative bacteria, which are negative, that will be communicate with, they will be connected with the chloride ion, and those which are positive, they connected with the crystal violet solutions. So now the iodide sample, this is the negative ion bacterial solutions. They will connected with the gram crystal violet, moderate interact, and large complex formations that will be inner wall of this. When the decolorizer will be using, that means stone or alcohol are using, they interact with the lipid membranes. That is the main things you can understand here. This is the main things. Chloride ion, because the cell wall who are negatively charged, that means with who are negatively charged, they interact with the positive things, and who has the negative charges, that will be go for the inner walls. So they can be formation of the this thing, because gram negative cells have the lipid membranes more thickness of lipopolysaccharide, and they get dissolved during the alcoholic treatment, and this is the negatively charged. That's why they remove these lipid molecules. Decolorizing agent, when you are using the decolorizer, this complex what formed by the negative charges and positive charges, that will be removed, while the positive charges, chloride ion, that is means of crystal violet, they are interact with the inner walls. They are not be going, because this inner walls wall was closed, because the decolorizing agent are the alcohol, acetone, they are dehydrating agents, so pores are become closed, so the inner walls there is no any effect. That's why the color of the crystal violet purple come after the counter stain also. So they are showing that the bacteria who are the less amount of lipids, that will be the positive, and who have the more amount of lipid in their cell wall, they are the gram negative bacteria. After using their counter stain, that is saffronon. Yeah, the basic question, these are the main things that can we differentiate the bacteria from positive or negative things with using of this gram staining methods. And this process we will tell you details about how can we making the, when you are going to take a slides, we make the stains for this gram positive negative things. We will put on them some amount of the culture on them and go for the dryness and put all these solutions one by one. One is the primary stain, that is the crystal violet, then we go for the second, this is the main thing, moderate iodide, then go for the alcohol, that is the decolorizing agent. After the washing them, you can remove this all thing washing and counter stain you using. Like this you will find out the properly dryness, and you will 30-40 seconds go for the again washing distilled waters, then you will see this type of the bacterial morphology, and where you can find out under the microscopes and see who are the gram negative bacteria, who are the gram positive bacteria. So this main things you can differentiate bacterial morphology on the basis of using different type of the process steps by gram staining methods. So some bacteria who are the gram positive, gram negative, I am mentioning here. You can see these are the bacillus, all the bacillus bacteria. These are mostly the gram positive bacteria: Nocardia, Clostridiums, Actinomycetes, Enterococcus, Corynebacterium, Listeria, Lactobacillus. Most of these are Mycoplasma, Staphylococcus. Most of the antibody producing bacteria are the gram positive bacteria, while the gram negative bacteria are the Escherichia coli, Helicobacter, Heliophilus, Neisseria, Klebsiella, who are the most of the infection causing bacteria are the gram negative bacteria. I will tell you why the gram negative bacteria are more infectious than the gram positive bacteria in the upcoming videos. Thank you so much for giving your attention to watching my videos YouTube channel. I would like to please subscribe and share with your friends who really like to interaction with this type of information of the immunology, microbiology. In coming days I will make on the video on these sections, so very not only for in this section, and also on to biochemistry and cell biology also. So thank you for again watching, and please subscribe and follow my Instagram page for your instant information of anything what you really